Hello, welcome back everyone. So daily we are studying media medical, so it's very boring to study a single subject, right? So today we'll be seeing about physiology, that is the plasma proteins. Okay, so let's start without wasting any more time. So in this chapter we'll be studying about its introduction, normal value, separation, properties, origin, functions, plasmapheresis, variations in plasma protein 11, and its applied anatomy, physiology. Okay, so I think you can see the screen. Okay, so plasma protein are serum albumin, serum globulin, and fibrinogen. Serum contains only albumin and the globulin, and fibrinogen is absent in the serum because it is converted into fibrin during blood clotting. Because of this, albumin and globulin are usually called as serum albumin and serum globulin. Because they contain serum, so they are called so. So the normal value is 7.3 gram per deciliter. Total proteins. Serum albumin is 4.7 gram per deciliter. Serum globulin is 2.3 gram per deciliter. And fibrinogen is 0.3 gram per deciliter. Albumin globulin ratio. It is the ratio between the plasma level of albumin and globulin is called the albumin globulin ratio. It is an important indicator of some diseases involving liver and kidney. So the normal ratio is 2:1. 2 is the albumin and 1 is the globulin. Separation of the plasma proteins. Various methods are available to separate the plasma proteins. So the well-known methods are precipitation, salting out, electrophoretic, Korn's fractional, ultracentrifugation. So precipitation method. Proteins in serum are separated into albumin and globulin by precipitation method. So in precipitation method, proteins in the serum are separated into albumin and globulin. Proteins in serum are separated into albumin and globulin. It is called a precipitation method. So in salting out method, serum globulin is separated into two fractions called euglobulin and pseudoglobulin. So in salting out method, serum globulin is separated into euglobulin and pseudoglobulin. Next is the electrophoretic method. So in this method, proteins are separated into albumin, alpha globulin, beta globulin, gamma globulin, and fibrinogen. Okay, so here the protein is divided into albumin, alpha globulin, beta globulin, gamma globulin, and the fibrinogen. In short, I'm telling, okay. So here it is given very briefly, so I don't read of this much because it is not important. Just the functions applied is important. Okay. Next is the Korn's fraction precipitation method. So in this, the plasma proteins are separated into albumin and different fractions of globulin. So in this Korn's fractional precipitation, the plasma proteins are divided into or separated into albumin and different fractions of globulin. Next is the ultracentrifugation method. So here albumin and fibrinogen are separated and their molecular weight is also determined. So here albumin and globulin and fibrinogen are separated and also their molecular weight is determined. Gel filtration chromatography, here all types of proteins are separated. So the process by which all types of proteins are separated is the gel filtration chromatography. Immunoelectrophoretic method, here quantitative method of proteins is done. Okay, so this much is enough to know about the separation. So next is the important one is the properties. Okay, so first molecular weight, albumin 69,000, globulin 1,56,000, fibrinogen 4 lakhs. Albumin 69,000, globulin 1,56,000, fibrinogen 4 lakh. So this is the molecular weight. And onconic pressure. Plasma proteins exert oncotic or osmotic pressure in the blood. Normally it is about 25 mmHg. Plasma protein exert oncotic or osmotic pressure in the blood. It is normally about 25 mmHg. Albumin plays a major role in exerting oncotic pressure. So here albumin plays a major role in exerting the oncotic pressure. Specific gravity is 1.026. Okay. Specific gravity is 1.026. Buffer action is the acceptance of hydrogen ions. Buffer action is the acceptance of the hydrogen ions. It is known as the buffer action. So buffer action is the acceptance of the hydrogen ions. Plasma proteins have 1/6 of total buffering action of blood. Okay. So plasma proteins have 1/6 of total buffering action of blood. Next is the origin of plasma protein. In embryonic stage, plasma proteins are synthesized by mesenchyme cells. In embryonic stage, plasma proteins are synthesized by mesenchyme cells. The albumin is synthesized first and other proteins are synthesized later. So the first produced is the albumin. So first albumin is synthesized and then other proteins are synthesized. In adults, plasma proteins are synthesized mainly from reticuloendothelial cells of liver. So in embryo, plasma proteins are synthesized from mesenchyme cells, but in adult, plasma proteins are synthesized from reticuloendothelial cells of liver. The plasma proteins are synthesized also from spleen, bone marrow, disintegrating blood cells, and general tissue cells. Gamma globulin is synthesized from B lymphocytes. So functions. Plasma proteins are very essential for the body. So following are the functions, that is role in coagulation of blood, role in defense mechanism of body, role in coagulation of blood, role in defense mechanism of body, role in transport mechanism, role in maintenance of oncotic pressure in blood, role in regulation of acid-base balance, role in viscosity of blood, role in erythrocyte sedimentation rate, role in suspension stability of RBC, role in production of Trephine substance, role in reserve proteins. Okay. So first is coagulation of blood. Fibrinogen is essential for coagulation of blood. Fibrinogen is converted into fibrin threads. Fibrinogen is essential for coagulation of blood. Role in defense mechanism. Gamma globulins play an important role in the defense mechanism of the body by acting as antibodies. So this gamma globulins act as antibodies and help in defense mechanism. Immune substances. Starling's hypothesis. Osmotic pressure exerted by plasma proteins plays an important role in exchange of various substances between blood and cells through capillary membrane. According to Starling's hypothesis, the net filtration through capillary membrane is proportional to hydrostatic pressure difference across membrane minus oncotic pressure. Osmotic pressure plasma protein exerts an osmotic pressure. Plasma protein exert an osmotic pressure എന്നിട്ട് എക്സ്ചേഞ്ച് ഓഫ് വേരിയസ് സബ്സ്റ്റൻസിന് ഇമ്പോർട്ടൻ്റ് റോൾ പ്ലേ ചെയ്യും. സബ്സ്റ്റൻസ് ഏതിനും തമ്മിൽ ബ്ലഡിനും സെൽസിനും ഇടയ്ക്ക് ട്രാൻസ്പോർട്ട് എക്സ്ചേഞ്ച് ചെയ്യാൻ വേണ്ടിയിട്ട് ഒരു ഇമ്പോർട്ടൻ്റ് റോൾ പ്ലേ ചെയ്യും. ഇതിൽ കൂടെ കാപ്പിലറി മെംബ്രെയിനിൽ കൂടെ ബ്ലഡിനും സെൽസിനും എക്സ്ചേഞ്ച് വേരിയസ് സബ്സ്റ്റൻസ് എക്സ്ചേഞ്ച് ചെയ്യാൻ വേണ്ടിയിട്ട് ഈ ഓസ്മോട്ടിക് പ്രഷർ എന്നിട്ട് പ്ലാസ്മ പ്രോട്ടീനിൽ ഒരു ഇമ്പോർട്ടൻറ്റ് റോൾ പ്ലേ ചെയ്യും. സോ അക്കോർഡിംഗ് ടു സ്റ്റാർലിങ്സ് ഹൈപ്പോത്തസിസ്, നെറ്റ് ഫിൽട്രേഷൻ ത്രൂ കാപ്പിലറി മെംബ്രെയിനിൽ കൂടെ ഉള്ള നെറ്റ് ഫിൽട്രേഷൻ എന്നിട്ട് പ്രൊപ്പോർഷണൽ ടു ഹൈഡ്രോസ്റ്റാറ്റിക് പ്രഷർ ഡിഫറൻസ്. അതായത് നെറ്റ് ഫിൽട്രേഷൻ കൂടിയ ഹൈഡ്രോസ്റ്റാറ്റിക് പ്രഷർ കൂടും കുറയും ഒരേപോലെ അക്രോസ് മെംബ്രെയിൻ മൈനസ് ഓൺകോട്ടിക് പ്രഷർ ഡിഫറൻസ്. ഹൈഡ്രോസ്റ്റാറ്റിക് പ്രഷർ ഡിഫറൻസ് across membrane minus oncotic pressure difference is the net filtration rate. Role in regulation of acid-base balance. Plasma proteins, particularly albumin, play an important role in regulating acid-base balance in blood. This is because of the virtue of their buffering action. Plasma proteins are responsible for 15% of the buffering capacity of blood. Plasma proteins, particularly the albumin, plays an important role in regulating the acid-base balance in blood. This is because of, it helps in the balance of acid-base balance through buffering action. Buffer action means acceptance of hydrogen ions. Plasma proteins are responsible for 15% of buffering capacity. So the plasma proteins are responsible for 15% of buffering capacity of the blood. Role in viscosity of blood. Plasma proteins provide viscosity to the blood, which is important to maintain the blood pressure. Albumin provides maximum. So albumin provides maximum viscosity than other plasma proteins. Role in erythrocyte sedimentation rate, ESR. Globulin and fibrinogen accelerate the tendency of Rolex formation. Rolex formation means piling up of the RBCs like coins. So globulin and fibrinogen accelerates, speed ups the process of Rolex formation by the RBCs. Rolex formation is responsible for ESR, which is an important diagnostic and prognostic tool. Role in suspension stability of RBC. During circulation, RBC remains suspended uniformly in the blood. This property of RBC is called suspension stability. Globulin and fibrinogen help in suspension stability of the red blood cells. Role in production of triphone substances. Triphone substances are necessary for nourishment of tissue cells. So, triphone substances are necessary for nourishment of tissue cells. Triphone substances are responsible for nourishment of tissue cells in culture. So these substances are produced by leukocytes from the plasma proteins. Role as reserve proteins. During fasting or inadequate food intake or inadequate protein intake, plasma proteins are utilized by body tissues as the last source of energy. Fasting or inadequate food intake or inadequate protein intake, plasma proteins are utilized by the body tissues as the last source of energy. Plasma protein is the last source of energy in case of absence of food. Plasma proteins are split into amino acids by the tissue macrophages. Plasma proteins are split into amino acids by the tissue macrophages. Amino acids are taken back by blood and distributed throughout the body to form cellular proteins, protein molecules. Because of this, the plasma proteins are called reserve proteins because it is reserved and is given or supplied in the case of absence of food. Okay. So that's all. Hyperproteinemia, dehydration, hemolysis, acute infections like hepatitis, respiratory distress, excess of glucocorticoids, leukemia, rheumatoid arthritis, alcoholism, hypoproteinemia, diarrhea, hemorrhage, burns, pregnancy, malnutrition, starvation, cirrhosis of liver, chronic infections, hyperproteinemia, acute infections, hyperproteinemia. Okay, so this chapter is completed. Next is the very important chapter, RBC. So you can see about RBC next class. Until then, ta-ta, bye. Thank you. So keep supporting, like, share, subscribe, turn on the notification.